Hi everyone. Okay, bear with me. As you all know, we are trying to scramble and figure out how to put things online. It's gonna happen. We'll figure it out. Don't panic. This is the third time, actually fifth time I've recorded this lecture. I'm trying different media to see what works best for you. So let's go ahead and get started. What my plans are is to go over this information. This is what we covered on Monday. And then we will go over what we covered on Wednesday. And then ideally you should be set for the quiz. Because I've been doing this, I've not been able to upload your quiz scores. Yes, y'all get 2 extra credit points. Yay, technology. Anyway, but I will make sure you're not penalized for any of this, and it depends on how long it takes for me to get this loaded, and then we'll just kind of go from there. Worst case scenario, let's just see what happens. So if you will recall, last time we left off on why be multicellular. So again, to be multicellular, that means you are made up of multiple cells. And we also discussed why there are some advantages to being multicellular. The first, of course, is division of labor. Being able to divide labor amongst other cells means that whatever you are doing, you get very good at. And then additionally, another bonus is that you increase in body size. Being made up of multiple cells, you get to be larger, which means you have a less chance of getting eaten. And so both of those things are good, okay? And they are definitely why it's an evolutionary step forward to be multicellular. Okay, so we also discussed on Monday what is known as the colonial theory of multicellularity. This suggests that animals are monophyletic, and remember, we talked about this long ago. You guys already know this. Monophyletic means the common ancestor and everybody from them. Okay, and so we also know that multi-celled critters evolved once the cells were coming together to live in colonies. And as I said a second ago, that means you've got a division of labor, so you get very good at the jobs that you do. Additionally, cells become dependent upon one another. Now, once that happens, that means that you're truly multicellular. Okay, so your cells are depending upon each other and they can no longer live by themselves. And then at this point, that's when you have what's known as true multicellularity. So, as we said on Monday, our first creature to kind of bridge the gap between multi-celled and single-celled are known as the choanoflagellates. Okay, so at the bottom left of this slide, you see an individual choanoflagellate. They're made up of the body and the flagella and all that other good stuff, and they can live by themselves. Or the other possibility is they live in a colony, okay, where you see several of them that are kind of clustered together on a stalk. That is also a possibility, and one advantage is working together. They might be able to draw a current and find food, which brings us to the peripera, otherwise known as the sponges. Okay, so the sponges are they're in the phylum Peripera, known as pore bearers. They have choanoflagellates inside of them, but they also have other types of cells. Okay, so they have some that actually provide structure, spicules, other cells that are sort of the good outside protective cells. They have multiple types of cells, and so this is why that choanoflagellates, because they're in there and they're in the sponge, the choanoflagellates provide the current, by the way. They use their flagella to provide a current. And so, but this is sort of what bridged the life between, being single-celled and then multi-celled and then eventually animal life, just like SpongeBob, although most of you are probably too old for SpongeBob, but that's okay. remember, those are what's generating the currents because they're using their flagella. So let's talk about how sponges develop. They start out with a zygote, and remember that's the fertilized egg. Okay, and then they go to the stage where they're free-swimming larvae. This is where they kind of decide where they're going to exist. All right, and then they will turn into an inverted larva. By this point in time, they're landing. Okay, and then they're completely sessile, where they will park it for the rest of their days. Once a sponge is an adult, it can't move around, so whatever environment is there, it has to be able to deal with. And again, if you look at a cross-section of the sponge anatomy, okay, you will see that you've got the spicules, you've got the choanoflagellate cells, okay, that they're providing the current. You have the other different types of cells, and each of them performs a certain job, and so that is how the sponge becomes successful. So let's talk about what it means to truly be multicellular and what you have to be multicellular. So in order to have what's known as complex multicellularity, this means you've got cell specialization. Okay, and that makes sense. We've said that before. Cell communication. Your cells, your body cells, all talk to one another. Okay, so don't forget that. That's how they know what to do their jobs. That's how they know what they're doing, that type of thing. And last but not least, adhesion. Your cells have to stick together. If they don't stick together, then you're not truly multicellular, and this arrangement is not going to work. Okay, so what I would like us to discuss next is when those instructions for cells to differentiate actually happen. Okay, so cells are basically told, what they're going to differentiate into, and this is evolutionary development is the basic field of science that studies this. Okay, and so what I want you to realize as your cells are growing and dividing, in most critters, actually all critters eventually, their cells will be told what they're going to differentiate and turn into. Okay, and this entire field is known as Evo Devo. I remember when it was coming out and everybody thought it was the coolest name, and I was kind of rolling my eyes because that's what I do. But just realize that 2 times cells can be told what to develop into. Okay, so the first is when the female lays the egg, and that should make sense because once she lays the egg, then genes are going to kick on and off based on those instructions that they receive. All right. The second time that cells can be told what to develop into is once the egg is fertilized. Okay, so once it becomes a zygote. So this should kind of make sense because once it's fertilized, then the cells get instructions on what they should eventually turn into as well. Okay, so again, what I'm wanting you to get out of this is cells are told, to develop into different parts twice, potentially. The first time could be once the egg is laid down, and the second time is once the zygote is fertilized. Okay, so one of the coolest parts about evolutionary development happens to be what is known as the Hox genes. Okay, and so the Hox genes are basically responsible for our cells developing into our different body parts. Okay, so the Hox gene, they're known as families, and I'll explain why in a second. But what I want you to get from this is that all animals have these Hox genes. That's first and foremost very important. So we have them, fruit flies have them, worms have them, mice have them, everything has Hox genes. This is what tells our body parts to develop into different things. Okay, so it's really important to know. Secondly, they all have similar instructions and similar locations on chromosomes in every single critter. So those in blue, as they are listed on the left, help with head development. Those that are in green are listed on the right, help with tail development, as you see in the slide. And those that are in the middle, okay, for example, in the red, help the middle of the body to develop. And so again, studying these Hox genes and how they differentiate and so forth is evolutionary development, okay? And I'm going to, in a minute, insert a slide or a video, I should say, and it's a pretty fun one to watch. It's completely cheesy, but I loved it. I wanted to share it with all of you guys because it's cheesy and fun. Don't panic over it, but it's a nice way to sort of visualize all of this information, and you get to hear it in a song, okay? So seriously, how cool is that? Okay, everyone, how cool is that to actually have an a cappella version of evolutionary developmental biology? I mean, I had to share. I just couldn't not. Okay, so hope you enjoyed that. Okay, don't stress about the details, but I thought it was just a nice way to sum up in a very cool way what we've been talking about. Okay, so now the next thing I want you to type in your notes, like right this very second, right, however you want to do it, is that these are the 3 different body plans of animals. 3 different types of body plans in animals, okay? The first is the sponge, just like we've been talking about. Okay, so these are multi-celled critters, but the thing you have to remember is they have no specialized tissues. This is why you can actually take a sponge and you can then pulse it in a blender, as we discussed briefly in class, or you can force it through a filter where you actually separate the cells again to get them to be single-celled. You let the solution sit and a sponge will form again in that solution. Okay, and so the reason for that is to, is that the sponges do have specialized cells, but they don't have any specialized tissues. So that's what I want you to gain from that. Now, another body type is known as the cnidarians. So these are like the sea stars, the anemones and the jellyfish. Okay, so the cnidarians are a very interesting batch. They have basically a sac-like body, and they have a mouth. Okay, so the mouth can be open and closed, and that's where food goes in and out. Okay, but that's kind of basically it. They're not super fancy, but you don't really have to be. They're functioning just fine, and they do quite well. Last but not least, we have our worms. Now, these are the more complex body plan, and by the way, this is where we fit. Okay, so the next time you guys are walking on a sidewalk after a rainstorm and you happen to see the worms across, remember those are our wormy little ancestors. They gave us our body plans. We're very grateful for that. So just ever so gently pick them up and usher them to the side. Okay, so the idea with worms is that they are basically advanced metazoans. Metazoan just means multi-celled creatures. Okay, and they have a fluid-filled cavity called a coelom, and we have that too. Okay, so we have a coelom, and that's what houses our organs. So we like that. Okay, then as we'll discuss in a few slides, they've got that fluid-filled cavity, but then they have a mouth, and then it consumes the food, and it passes through the anus, just very much like us. So, but this is definitely where we got our early body plan from. So the next thing to remember is that sponges, as we've said, they're in the phylum Porifera. So remember, phylum is kingdom, phylum, class, order, family, genus, species. King Philip came over from Greece Saturday. So this means that they are the phyla called the pore-bearers, okay? And again, as we said, they don't have distinctive tissues. Now, these are not loofah sponges. Those are actually completely different. Those come from plants. And then actually in the next slide, I'm going to show you, because it's still pretty cool to know, where the loofah sponge comes from. But these are the actual sea sponges that live in the ocean, and this is where they, from a classification perspective and the characteristics they have, this is where they belong. Okay, so just as the interesting side note, this is where loofah sponges come from. Now, these are not the sea sponges that we use, okay, or that we've been talking about. However, I like to throw in information when I find it interesting, and so I plan to grow some this spring. We'll just see how it goes. Now, as we're classifying our critters, there are two different types. There are what are known as diploblast and triploblast, and these are, again, fitting in the evolutionary classification. So, we've said this before, di means two. Okay, this means that they have two tissue layers. Okay, so we're away from the sponges down, we're actually talking about tissue layers. Triploblast, tri means three. Okay, so they have three tissue layers. So two tissue layers, we would be talking about jellyfish or anemones, and they have what are known as the ectoderm. Ecto means outside, okay, as well as the endoderm. Endo means inside. All right, so the diploblast, that's all they've got. Now, a triploblast, and by the way, we are triploblasts. In addition to the ecto and the endo, there's also the mesoderm. Now, remember, meso means middle. Okay, and ultimately, this will develop and house our internal organs. Okay, so that's what the mesoderm will develop into, and just gives you an idea of what, we're considered more evolutionarily advanced. All right, whereas the sea sponges and cnidaria, not the sea sponges, the sea anemones and the jellyfish are considered the diploblasts and more primitive. Okay, so we also, in addition to being diploblasts and triploblasts, we are classified based on our symmetry. Okay, so radial means just like a jellyfish, you can cut this individual any different number of ways, and technically you will still have mirror images, or at least close enough to it. All right. Now, that's not us. We have what's known as bilateral symmetry, okay? And the more evolutionarily advanced critters tend to have the bilateral symmetry, and that's when you can only cut us down one plane and still have mirror images, okay? And so, just realizing that, we're the bilateral plus we're triploblast, okay? The jellyfish are radial, and they're also diploblast. So let's talk about those body cavities, okay? Because there are exceptions in nature, because nature basically does what nature does, and we just try to classify it so we understand it better. However, there's always exceptions to every rule. Now, when it comes to body cavities, there are critters that are known as acoelomate. Okay, so those are like the little platyhelminthes that are on the left. A means not having, and coelom means body cavity, so they don't have a body cavity. Okay, so that's what acoelomate means. Pseudocoelom, pseudo means false. Okay, so false body cavity. And that's what the middle creatures, those would be like the roundworms and so forth, have. And the best way I can think of to explain this to you is to actually make a direct comparison to the coelomates, which have the true body cavity, which is on the right. Okay, so if you look at the coelomates with the true body cavity, you will see that the body cavity is completely surrounded in purple, and that's the mesoderm. Okay, so you can see it all the way around on the inside and the outside is surrounded by purple. Whereas the pseudocoelom, on the very, very inside of that body cavity, it's the endoderm, which is represented in yellow. That's the difference. Okay, so pseudocoelom, not completely surrounded by mesoderm. Coelomate is completely surrounded by mesoderm. And then again, acoelom means they don't have a body cavity at all. Okay, so as we're talking about our fellow coelomates and our little worms, what I would like you to consider is to think back to grade school or high school when you guys dissected your first worm. I'm not going to lie, I hated it. I didn't do well with dissection, so I went into plant research. However, the thing to realize, though, is that worms are very much like us. They've got a mouth, they've got our hearts, they've got an esophagus, blood vessels, intestines. Okay, they've got an anus. They eat and process food much like we do, okay, which is why they fit into the same classification as we do. And, of course, you're not expected to memorize this, but what I want you to get out of it is they share a lot of the same traits as us. Therefore, you can see why we're related. Okay, so what I love about this slide, there's a lot of detail on here, but you should not panic, okay? What I want you to realize, though, is it does a great job of summing up the evolutionary history of our multicelled animals so far. So at the very bottom, you got the ancestral choanoflagellates, okay? So we've talked about those guys. They can either live single-celled or multicelled. Then we get into our porphyrians, or our sponges, okay, which don't have true tissues. Then you get to the which means true metazoans, tissues, okay, true tissues. And so then that splits into the radial symmetry, which is to the left, and the bilateral symmetry, which is to the right. And then you get to what is known as the protostomes and deuterostomes, and this is what we're going to talk about next. So the protostomes and deuterostomes, for those of you that were in lecture on Monday, you will recall that this is basically embryological distinguishing, okay, based on embryologic patterns. And so we are deuterostomes. The simpler critters are protostomes. We split about 540 to 670 million years ago, okay? And we will talk about the characteristics that divide us into the two groups, and basically it's just what certain cells turn into from starting with an embryo, and everybody in each of those groups, will have certain characteristics. So let's talk about the protostome and deuterostome divisions. Okay, there are 3 different ways that protostomes and deuterostomes are divided. And so I do want you to be able to explain these. So the first way is through cleavage. Now, cleavage is nothing more than how the cells divide. Okay, in the protostomes, at the 8-cell stage, you'll notice that the divisions are not even. So it's known as spiral when they're not even. Okay, so the ones at the top of the cell are smaller than the ones at the bottom. They are also what's called determinate. Determinate means at this stage they know what they're going to turn into. Okay? The next thing to pay attention to is the deuterostomes, because they are different. They have what's known as radial cleavage, which means the top and the bottom cells are divided evenly, and also indeterminate. And this should make sense because they don't know what they're going to turn into yet. Okay? So again, when it comes to cleavage, protostomes, it's spiral and they're uneven, as well as determinate, which means they know what they're going to be. The deuterostomes at the 8-cell stage are radial, which means they're basically evenly divided, and indeterminate, which means they don't know what they're going to be. Okay, so that's the first way protostomes and deuterostomes are distinguished. Now, the second way is how the body cavity forms. Remember the coelom. Okay, so now we talked about this in class, that your cells grow and divide and grow and divide. Okay, is how they happen in the embryo. Then eventually what happens is that your cells all move to the outside, okay, so it's a hollow ball of cells. This is just the way embryology works. And then what happens, it starts to invaginate down below, and so that's called the blastopore, where that kind of pushes inward. Okay, and you see that at the very bottom of those particular drawings there. That's the blastopore. So the first thing to pay attention to is that the coelom forms in protostomes. At this particular stage, the coelom is already pinched off. Okay, so it's a separate batch of cells. Whereas the deuterostomes, the coelom, now remember the coelom is in red in these two diagrams. And so in the deuterostomes, the coelom is still attached, so it's up at the top. Okay, I know that sounds nitpicky, I know that sounds complicated, but remember we're dealing with just how cells form and embryos, and as you may have recalled, I said in class, every little stinking batch of cells has a name. And so this is just something to keep in mind. This is the way it's done. Okay, now the third way protostomes and deuterostomes are differentiated is based on what the blastopore turns into, or that first invagination, as we said a second ago. So the protostomes, it turns into the mouth. Okay, the deuterostomes, what happens is the anus develops first. And just a reminder, because I cannot resist the joke, we're deuterostomes. Okay, so what I want you to get from the slide is to be able to distinguish the three different ways that protostomes and deuterostomes are differentiated based on how the embryo is developing. So let's talk about what we have discovered so far. And by the way, you all should be adding this information to your timelines. I totally forgot to mention that with all of the fuss and everything that's going on. So you should be, building upon your timeline. You're going to turn it in at the end of the semester, and this information should be there. Okay. So the Earth was formed 4.6 billion years ago. The oldest known rocks we know are somewhere around 3.8 billion years ago. Oldest known fossils, as you see. We know that we had the transition to oxygen atmosphere. Now think back when we were talking about banded iron formations, or BIFs, okay? So remember, that's when the Earth alternated back and forth between having oxygen, meaning the iron precipitated out, and not having oxygen, meaning the chert was the sediment. And that would go back and forth between iron chert, okay, which showed that the transition to oxygen was not immediate, so it took a while, okay? And so there was no oxygen early on, or not much. Then you had the transition to oxygen atmosphere, most likely due to the single-celled photosynthesizers, and then afterwards you only had rocks that could form in the presence of oxygen, okay? So be thinking about that as we're going through this information. Eventually the eukaryotes would come about, okay, and then we would have what I like to think of as fashionably late entrance of the animal fossils. Okay, so that's when our ancestors would finally decide to come around. Okay, so the next thing I want to talk about is the actual fossils of these early critters and where they were found. And I'm sorry that the slides, the writing is a little bit off. I'm not sure what happened, probably something in the translation. But the earliest fossils and evidence of life are found in the White Sea of Russia, the deserts in Africa, and the Ediacara Hills of Australia. Okay, so this is where our earliest animal fossils are found. Now, some of the earliest scientists to work on these early critters, Reginald Spring, Adolf Seelacker, Martin Glassner. No, you don't have to know their names. I always just like to show them to give them credit for what they were discovering. And what these guys were wondering is whether or not the earliest animal fossils gave rise to who exists today, or if they died out and who we have today came about in basically a second evolutionary trajectory. And so ultimately I'm going to show you a video with David Attenborough. Haven't figured out how we're going to do this yet because I might have to upload the whole video, and that's going to be a while. So we'll see how this goes. I'm not sure how to have you, we'll figure it out in a second. But you'll see that they were arguing over the fossils that they found during this time. Are they the ancestors of everybody that exists today or not? Okay. Now what I want you to get from this, again, you don't have to know their names, but what I want you to pay attention to are the critters that existed and sort of what the evolutionary patterns are that are going on. Okay, and so that will also be emphasized in the video that I will have you guys watch too. So in this next slide, I'm wanting you to pay attention to what these early animals look like. Okay, so to realize first off, they're pretty small, somewhere between the size of a penny and maybe a dinner platter at most. Okay, they have radial symmetry. You will see that here. They also have, as they have radial symmetry, they have true tissues. Okay, now these kind of look like jellyfish fossils, but in order for them to preserve like this, they basically would have to flip over perfectly every single time, and I'm not really sure that's the case, but they are hypothesized to be related to early jellyfish. You got to remember that when it comes to these fossils and so forth, all you have is what you can see in the rocks, and so your inferences are a bit limited when you're trying to figure out if they're related to anybody that exists today, so you just got to do the best you can. So remember also, I'm not going to ever on an exam or quiz flash a creature and say, who is this? That would be hideous, okay? But what I do want you to do is to realize, what the different traits are that are going on and maybe list some examples, because that would totally be fair game since your quiz is going to be open note, open book, all right? So if you look at the critters we see here, they look to have some interesting traits. It looks like they have bilateral symmetry, maybe a head and a tail, maybe some segments, okay? So they definitely have some interesting things going on. They are still pretty tiny, don't forget that, okay? However, they're definitely becoming more evolutionarily advanced. So one of my favorite groups is known as the Dickinsonia. These guys are so cool, okay? They, again, could be the small as a penny, size of a dinner plate. They have segments. You guys can see segmentation. Hints of bilateral symmetry. You definitely can't, cut these guys and have mirror images all the way around. So it's starting to get to that point. The other thing I forgot to mention earlier is so far everybody just has soft parts, okay? So there's no bones and no hard parts yet. So as we think of evolutionary trajectory and where everything is, remember that as well. Okay, so just take a look at these guys. They might potentially be some sort of polychaete, some sort of worm. It's just really hard to tell. But what we do know is that, they're starting to gain some interesting traits. Now, we've talked about this before as well. So, the fossils that we find for these critters, there are also what are known as trace fossils. And see, the stuff comes back and it all ties together. We talked about trace fossils before, so those are footprints or those are tunnels, okay? Evidence left behind by the critter without actually being a fossilized version of the critter. Okay, so there are lots of trace fossils of these guys, and the thing I want you to get from this is that means they are moving around. So as you're thinking about what evolutionary traits are going on, consider that they're also moving around in addition to having some radial symmetry, some bilateral symmetry, some segmentation, and all sorts of other good stuff is evolving at this point. Okay, another favorite group of mine, the Tribrachidiums. Look at this. These guys are so cool. Even today, we don't have many creatures that are composed of segments in threes or anything in threes, okay? Tri means three. Okay, so Tribrachidiums, these have segments of three, all right, which again is very rare. The only family or group of creatures that has things in threes today is the lily family. So Easter lilies are going to be coming out soon. You can count the petals in threes and the leaves in threes. So the next time you guys are at Meyer and you see the lilies or the day lilies, check everything out and count them. Count the leaves and the petals and you find that they are in threes, okay? But other than the lily family, I don't know of anything from an evolutionary perspective that has things in threes. So that's what makes these guys so cool, besides the fact they're really interesting looking. I mean, seriously, how cool is that? Okay, so we also have the next group, which is, I have to admit, I love all these guys, I guess. The Parvancorinas. So these guys look like early trilobites. You can see that they have a hard, looks like a T going across the top by their head. Then they have something hard going down their spine, okay? These look remarkably like trilobites, which are also one of my favorite group of creatures, okay? The trilobites that we talked about before that we know and love that died a long time ago, but they're still actually pretty cool. And so it's hypothesized that these guys are a distant relative of who would eventually turn into the trilobites. So the next group is the Spriginas. Okay, now initially the researchers weren't sure if Spriginas kind of stood on their head like that middle drawing, or if they moved along the bottom, which seems to be more of the consensus. But you can also see segments in these guys. You can see bilateral symmetry. You can see a head and a tail. Okay, so evidence of movement and all sorts of other cool stuff. They're still pretty tiny, all right, but they are really interesting and potentially could be an annelid worm or something along those lines or some distant relative. Again, just keeping in mind when all you have is a tiny fossil, it's hard to figure out who eventually evolved into who. Okay, so the next two critters, and again, just pay attention to the traits that are going on. Cloudina, which is something happened with the format of the slide, I'm afraid. The Claudina is the creature on the left, and the Maculathus is the one that's cool looking on the right. Okay, the Claudina, if you recall, remember the name Preston Cloud. That's who this creature was named after, and they're basically worms that have really hard tubes that form for protection. And then the Nemaculathus is the guy on the right, which kind of looks like a poppy flower, you know. I'm not sure how to really describe him other than he looks really awesome. And okay, so realizing that these are the guys that existed, and again, they were pretty tiny back then, so life was definitely relatively small. Okay, everyone, so the only issues I'm having with this program so far is being able to only show you clips of a video rather than the whole thing. Okay, and that is a challenge. Now on the next slide, there is a video that you guys should be able to play. I would like you to watch the whole thing. It's about an hour long. I know it's long. Everybody's, I'm sure you're all giving the computer the look, okay? But it would be good for you to actually watch the whole thing. And plus, it's David Attenborough, who, let's face it, is awesome. But however, if you really are crunched for time, although you should be able to still be able to fit this in, you can just pick up at the end, starting with the last half hour. So start after it's been for 30 minutes and watch till the end. And if you can do that, you will have all the information that you need. So before I forget to, all of us now, by the way, are messing with different formats for the videos, trying to figure out what works. I told you early on, this is the 5th time I've recorded this lecture because the other platforms are not functioning properly. What I would like from you, though, is feedback. So were you able to watch this? You know, could you download it okay? Did everything play okay? Can you hear me okay? All right, so please do give me some feedback. You all know I take constructive criticism well. I want to make sure that you're having the best experience out of this possible, okay? And so if I can do anything to improve it, please let me know. So as always, stay safe. Wash your hands, use common sense. You're going to be fine. Everything's going to be fine. And I look forward to chatting with you in the next lecture. And I'll talk to you soon.